full day behind. So today we're going to run through, refresh your memory on basic cell division stuff. We'll get into meiosis and really talk about the details on that. We're talking conceptually about this, how things move here, how things move there. In the next section of the course, section 3 of the course, we get into the molecular mechanism of it. How does DNA polymerase get started? How does it copy the DNA? Nucleotides moving in and such. How does crossing over occur? We're giving a general discussion, so in the next next week we can talk about mapping of genes and we talk about linkage of genes. We'll work through the details on these. So, in bacteria, the process is pretty simple. We'll touch on this a little bit when we get into DNA replication, but the basic idea in bacteria, they are a great model organism. E. coli was a great model organism. What happens to replicate the DNA there is very similar to what happens when we replicate the DNA in humans. If we have our two DNA strands like this, as we talk about with PCR, the DNA has to somehow be separated. There's actually what we call a replication fork that occurs. There's a replication bubble, so the DNA polymerase can get in here and make a copy of this. So the copies get made, and ultimately what you're left with, because the genome of the bacteria is circular, you wind up with the original strand here and a copy. And you wind up with an original strand here and a copy. If you work the details out through the replication process, which we will in the next section, essentially what you get is the DNA, so this circle is connected to this circle and they're locked together. And there's going to have to be a way to break the sugar phosphate backbone for some reason. We're going to talk about this in the context of human cells also. For our cells, when we talk about DNA replication, in general, we're focusing on the nuclear DNA. We will actually talk about places where Mendelian genetics fails. So we talked about Mendelian genetics, and we talked about extensions to Mendel, incomplete dominance, these kinds of things, epistatic interactions. And there are some circumstances where Mendel's laws do not apply, and that's generally when you talk about inheriting mitochondrial DNA, chloroplast DNA, these types of things. So I want to introduce to you these concepts, this cytogenetics. So cytogenetics is basically studying the chromosomes as their own organelles, as their own kinds of structures. And there's a whole field devoted to this. It is possible to take a sample of blood from a person, or basically maybe there's a fetus, an embryo or a person growing in the womb of someone. We can take a sample of the fluid, the amniotic fluid from the woman's uterus, from the womb, and just like as you walk around, things are flaking off of you, you've got skin flaking off of you, that baby growing in there, cells are falling off of that baby, and they're in that fluid. We can actually take that fluid out in the cell and gather the cells out of that fluid. We can take those cells and we can actually look at the chromosomes, and we could diagnose this child has an extra chromosome 21. This child is trisomy 21, and we'll have Down syndrome and prepare the parents for it. Some people might say, hey, we need to abort this pregnancy. Some people would say, okay, great, now I can plan ahead and I can mentally prepare myself for the challenges. That's not my position to help you choose which way to go on that. It's your own decision. Cytogenetics is a field of studying these chromosomes. What we see in this image up here is all those little worm-like things. Someone has gotten the chromosomes of a cell, spread them out on a field, basically a glass slide, we'll say. And then they've been arranged, and this is what we call a karyotype. Chromosomes have been arranged. So this is chromosome 1, it's probably, that's for mom, and this is chromosome 1 for dad, or it's vice versa. I don't know, there's no way to really tell, but just by looking under a microscope. So the homologous chromosomes are lined up and laid out like this, and this looks to me like a normal karyotype and we'll say male is normal, okay? Today, we'll say male is normal because there's an X and a Y, okay? So, there's value in being able to look at this. It can actually be done pretty quickly. And most eukaryotes, oops, that's not it. That's the wrong button here. The process of actually preparing this is kind of tricky, and it's actually very much an art. So the cells are treated with a chemical so that they get stuck in metaphase. So the chromosomes are all condensed into that format that you'd be able to see in a karyotype. And then you have your cell, you've got your plasma membrane, and then the nuclear membrane, nuclear envelope, and the chromosomes are in here like this. All the chromosomes are in there condensed. The cells are treated in a way, basically, you take detergent, soap. The active part of soap is called SDS, usually, sodium didosyl sulfate. And that will wash away the lipids out here, but it's unable to wash away the nuclear membrane. So what you wind up with in a tube is a whole bunch of intact nuclei and a whole bunch of cell debris, which is the plasma membrane we washed away, and all the cytoplasm is floating around in here too. We can take this tube and we can spin it, and the nuclei will pellet on the bottom because they're big and heavy, and everything else will be on top. We take the liquid off top and get rid of all this stuff, and we're left with pure nuclei here at the bottom of the tube. Here they just show us blood cells. It could be nuclei, whatever, something large. And then this is the tricky part. This is crazy. They'll take a saline solution, a PBS, something that's the same osmotic concentration as your blood, right? And they'll put that in there, and they'll suspend the nuclei in this liquid, and they take the nuclei and have them in a dropper bottle, like an eyedropper, and they take a glass slide and they set that down, and they squeeze a drop out of the dropper, and the drop eventually drips and falls and splats onto the glass slide. And as it hits, the nucleus smacks onto the glass slide, and the nucleus splits open, and the chromosomes go splatting out into a chromosomal spread, like this. And if you're lucky, the chromosomes are all separated away from each other, and you don't have them overlapping with each other to make it a messy situation. You'd say, okay, well, I know that if I drop from this high, I can actually, over time, develop my technique. If I drop it from 2 feet, I don't get a good spread. If I drop it from 2.5 feet, I get a good spread. 3 feet, and the chromosomes all break apart, so you can measure this down. Unfortunately, that technique of dropping and getting a chromosome spread, literally dropping the nuclei, the ability of the chromosomes to spread out depends upon the barometric pressure, the humidity in the air. I don't know whether you shampooed your hair that day. I don't know. It's an art. And I can remember I was working in a lab and the guy was doing these chromosomal spreads of mouse chromosomes. And I was like, hey, aren't you dropping in a nuclei today? I wanted to watch how you were going to do this. He said, oh, no, it's too humid today. It won't work. Okay. It was too humid walking in. It won't work today. Okay, fine. So in the old days, and I actually had a college graduate friend of mine, a good friend of mine, they would get these spreads, and they would print them off on a piece of paper, 8.5 by 11 piece of paper. And her job, this is how old I am, her job was to take scissors and cut the chromosomes out from the paper, and take the chromosomes and line them up next to each other, and tape them onto a fresh piece of paper, the little cut out pieces on a fresh piece of paper so that she got this, and then they would lay that down and take a picture of that and give that to the doctor so the doctor would go, okay, so here's my karyotype of this. Nowadays, they have a computer program that goes, ta-da, look at that, and it arranges it all digitally in an instant. And the computer's able to do it because the chromosomes get different bands on them. So there's a stain that you can put on DNA, and it's called Giemsa stain. And this stains some parts of the chromosome dark, and other parts of a chromosome it doesn't stain very well. So what you wind up with are these bands, like this. So if this is chromosome number 1, X means chromosome, X-zome is chromosome, exon 1. If this is chromosome 1 from mom, if you took chromosome 1 from dad and laid it out next to it, the banding pattern would be the same. Except in this particular instance, somebody who's trained, so the banding pattern for your chromosome 1 is the same as my banding pattern. Every chromosome 1 stains the same way the heme sustain. And the pattern of bands is the same. Except in this particular case, I left, there's 1, 2, 3 bands here, and there's only 2 on this one here. So one of the reasons people do cytogenetics is, hey, this person seems to have an allergic reaction to peas. How strange is that? And there's 3 other cases that I'm aware of in the Denver area where people are allergic to peas. Well, why don't we do a chromosomal spread and compare the chromosomes of all these people, and if everyone who's allergic to peas is missing this 3rd band here, there must be a gene there that's involved in being tolerant of peas. Right? So we can start to say, oh, so now that we know, based on what you did in the NCBI project in lab, you know that that's the locus. And you now know you could go to that spot in the Human Genome Project and say, what genes are located in that area? And maybe there's 50 genes, or maybe there's 3 genes. And we start to map down and say, I'm starting to know the function of this gene, which maybe no one knows the function of that gene. Okay. So this is called a karyotype right here. And I know you guys can't see it very well, but I don't want to turn the lights off because it screws up the camera. Is anyone been watching the lectures on Facebook or YouTube? You type in genetics and CSM on YouTube. It's wonderful to hear that all of Katie's hard work is paying off.
We haven't had an exam yet, though.
Oh, you had one exam?
We had one exam, it wasn't that hard.
Yeah.
So maybe the next exam will be harder.
Well, I will step up to the challenge. I'll tell you next time. No, okay, yeah. No,
Actually, you teach us so well that there's no need to review it because it's all in our head the first time.
Great, I'm glad everyone is going to get an A next time.
Oh,
We'll see
yeah.
about that. Okay. So we could actually, we'll say blue, like baby blue is boy for little babies, right? So pink would be girls. So if we have different colored chromosomes, I'm going to say the moms, this came from mom and this came from dad, just for convenience sake. Is anyone offended and want me to do it the other way around? Okay, Katie, you don't get to vote, sorry. And oh, Orlando also, sorry. They both watch these. So here we have the karyotype, and the banding pattern is the same. But the DNA sequence may not be exactly the same. That's because there's different alleles. The reason there's a blue-eyed allele is the sequence of the gene that codes for eye color is different. How different? It's probably off by maybe like 1%. You have the brown eye allele, if we call that the normal allele because it's the most common. The sequence of the blue version of that is 99.5% the same. Right? But we can illustrate the different alleles on here. So this person has, if we call this chromosome 1 from mom and chromosome 1 from dad, they obviously they're diploid, they have 2N. This person is homozygous dominant for gene A. This person is a carrier, or they're a heterozygote for gene B. And this person is homozygous recessive for gene C, right? Okay, so these are homologous chromosomes right here. In lab, we define locus. Locus is a fancy way of saying the location on the chromosome. But that's a really long thing. Location on the chromosome is a long thing, so we made up a word called locus. And I have no idea why they chose that. Whereas bacteria divide by binary fission, they duplicate this, the chromosomes go to opposite poles, they build a cell wall in between, the cells go off and they do their own thing. These are genetically identical organisms, right? Who says they're genetically identical? Yeah, that's That's what we say in General Biology 1. What actually happens is as DNA is getting copied, that DNA polymerase, on very, very rare occasions, puts the wrong nucleotide in. And so maybe 1 in 1,000, or no, 1 in 1,000,000, we're going to say, nucleotides gets put in incorrectly. So although these are sister cells from one original parent, they do have DNA sequence differences, 1 in 1,000,000. Right? So probably up to this point in your college career, in your biology career, people would say all of the cells in you are genetically identical, and that's not true. Because you started as one cell, fertilized egg, right? When that cell divided, you have a nucleotide, you have 3.2 billion nucleotides in your genome. And if the polymerase makes an error, 1 in 1,000,000, this has accumulated its set of mutations, and this has accumulated its set of mutations. Even when you were at the 2-cell stage, you were not genetically identical, your cells. And so as those cells have divided, you've gotten a tremendous amount of genetic variability within your system, just you. You're a mosaic. Very, very closely related, very few differences in the nucleotides, but your cells are not the same. How cool is that? That's why I have this red asterisk here. So hopefully at this point in your college career you have talked before about the cell cycle. I'm going to run through this quick and then we'll get into meiosis. So we can actually look at this as a circle. I'm going to draw it originally as basically a biochemical pathway. And I don't know how many steps I'm going to need. I'm just starting with 4. We have a cell, and I'm not going to draw the nuclear envelope and all of this. And there's these chromosomes in here, and they're matching up like this. This is homologous for chromosome 1, and these are the homologs for chromosome 2. These chromosomes need to get copied. Then the chromosomes need to segregate to opposite sides. And then eventually we're going to pinch this shut. And we get 2 cells that are genetically identical, right? So this will separate into 2 cells. Like that. So we can draw it linearly like this, but this is basically this cell is basically this cell, and we can run through the cycle again. So we draw it as a circle. So as we draw this out as a circle, we start with a cell that is just divided. So one cell goes off and we're not going to worry about that cell, and the other cell we're going to think about. So this cell, actually, because it's divided, the volume of this cell is small because you've split all of the cytoplasm and all the material from this cell into 2 cells. So the cells are really small. If this cell tried to divide, you get even smaller cells. So there comes a point of time where a cell is so small, it doesn't have enough resources inside it to survive. Let's say this cell has 2 mitochondria, and it divides into 2 cells. What if both of the mitochondria wind up in this guy, and this guy doesn't have mitochondria? Human cells have to have mitochondria. For our purposes, they have to have mitochondria to survive. The cell would die. So there has to be a period of time where a cell which has just come out of cell division gathers resources and grows physically bigger so that it could actually divide again and its progeny would each get. And that process, that step is called G1 or growth 1. And once a cell has reached a critical size and it has enough sugar stores in it, and it's in a happy environment, there's a lot of vitamins and amino acids all around, it says, gosh, things are great, we can afford to have another cell. There's enough surplus in the environment to support a second cell. That cell will go through a very important checkpoint that I'm going to call start. A geneticist would use the word start. I'm a cell biologist. My natural thinking is to say restriction point, because apparently cell biologists are pessimists and geneticists believe the glass is half full, right? So once the cell passes start, it goes through DNA synthesis. And I misspelled synthesis. Synthesis, in other words. We call that S phase. And that is the process, that is the time when the DNA is being separated and physically copied, just like a PCR reaction. The time the DNA polymerase is running down nucleotides and making a duplicate. Once that is fully completed, the cell needs to turn off all the machinery that it used to copy the DNA synthesis. And it needs to turn on the machinery that's going to separate the chromosomes and physically divide the cell into two cells. And that is called gap. Some people used to call it growth. They thought the cell had to take up more sugars and get to a happy place again. Now it's just, so they used to call it growth 2, G2. And now people call it the gap phase, G2. The cell is switching gears from copying DNA to how we're going to separate and split myself in half. Once the cell has reached a certain point where it's able to do that, then comes the infamous mitosis, or M phase. This is prophase, metaphase, anaphase, telophase, cytokinesis. Some people put that in there as part of mitosis, right? So what is the term for all of this right here? This is prophase, metaphase, anaphase, telophase, cytokinesis. What is the term for all of this? Interphase, right. Where does DNA replication occur? Metaphase? Interphase? Let me ask that again. When does DNA replication occur? Interphase? Is it metaphase? Interphase. Sure it's not telophase? Or prophase? So, every year in this class, I make a serious point of stopping the class to tell everybody, to get everybody's attention. DNA synthesis happens in interphase. On the exam, I guarantee it, every year, at least one of you will tell me that DNA replication happens in metaphase. Even though we just did this, I do this every year, somebody in this class. At least one a year is going to do that. Well, please don't be that person. I'm telling you, I guarantee you this question is on the exam. Guaranteed. Please don't miss it. When does DNA replication occur? Interphase, not during mitosis. Got it? Okay. So I've gone through all of these details here. This start, why is start so important? Yeah, so you start replicating DNA. What's the big deal about that? This is actually a point of no return. This is like walking through a door and you close the door behind you and you're like, ah, crap, I forgot my house keys in the house. And you turn and the house's door is locked. There's no going back. Does that make sense? You ever shut the door in your car and you're like, oh, my keys are sitting on the chair. And your car keys are locked inside you. Why did I do that to myself? This is that same situation. It is a point of no return. Once a cell has moved past start, people have done this in lab. Once a cell has moved past start, if you take the nutrients away in the lab so the cell is in a not happy, healthy environment, the cell will try to finish, and if it is unable to, it will die trying. It will not go back and say, oh, false start. Somebody played a trick and took away all the sugar. Go back and we're back in G1. That's it, it can't. Once the cell has passed start, it will finish this it will finish and go back to this point or die trying. Okay. When we get into the mitosis is where we actually see these chromosomes come together in these shapes. This is pretty cool. This is someone who's done a scanning electron image of this. The DNA is so compacted it forms this amazing structure. Normally it's not like that. Where does this structure, I mean, although it's not, we see it condensed during mitosis, where does this actually occur? Where is that thing created? When? S phase, interphase, right. So early in S phase, you don't have this because the DNA is just getting started. But when you get near the end of S phase, if you were to look at the chromosomes, they look a lot like this. So all through G2, they're like this. They're just not compacted in a structure you can see it. They're diffuse all throughout the cell. But anyway, good. So what we've got here is these strands have separated through the process of DNA replication. And I wish they had done this. I wish they had drawn this a little differently. So this guy was, this strand here was copied. And this strand here was copied. Like this. So that this consists of one of the original strands of the DNA and one copy strand. And this is the other original strand and a copy of that strand, right? And these guys are tied together with a structure that holds these guys together, and that is the waist-like constriction here. And the sequence of DNA, this is a whole bunch of proteins that hold these guys together. We'll call these kinetochore proteins. And technically, most of these are given specific names and they're called cohesins. Because they hold things together. Don't worry about cohesin. Kinetochore proteins. It's a whole bunch of different genes that all participate to make this big mass of protein that holds these guys together. Those proteins have a consensus sequence that they bind to. Is that a surprise? And the consensus sequence is called the centromere. Okay? This sequence of DNA is called the centromere. If you cut this sequence out, if you cut that sequence out and you stuck that DNA sequence right here, and right here, oh, I should, sorry, that's bad. This is supposed to be here, right? Did you guys notice that? These are the two copies, my bad. I'll leave that up there. If I put the centromere sequence here, these guys would end with it here and end with it here. What you would get is a chromosome that looks like this. You would get two waist-like constrictions on this chromosome, where they're tied together, where kinetochore proteins are holding them there, and kinetochore proteins are holding them there. People have done that, and you isolate those chromosomes, and that's what they look like. Okay? So these guys are called sister chromatids. This is a chromosome. I want to define this. This is a chromosome. How many pieces of DNA, how many strands of DNA in that chromosome? 2. This is a chromosome. How many strands of DNA in this chromosome? 4. 4 strands. Oh, let me do this. We continue through the mitosis such that these things go away and these guys break off of each other. This is a chromosome. And this is a chromosome. How many strands in this chromosome? 2. How many in this? Right? If these guys are tied to each other by this protein bridge, they are a single chromosome. They are sister chromatids of one chromosome. The instant, that one instant in time where this finally breaks and they're finally separated from each other, now they're not sister chromatids anymore, they're homologous chromosomes. Okay? I'm going to make sure you understood those terms. Okay. I've gone through all this. So, I'm not going into great detail on the prophase, metaphase, anaphase, telophase. By now, I hope you had that in high school, you had that in general biology 1. I hope you've gone over that stuff. Just work on that as a base. I do talk about meiosis a little bit because we're going to talk about the recombination that occurs in meiosis. Okay. When the chromosomes line up, does anyone know the name of this thing? What's this overall structure? Centrosome, or is it a centromere? Huh? Centrosome. Who says centrosome? Who says centromere? You guys are total chickens. You have to vote. Who says centrosome? Oh, okay. Who says centromere? Yeah, so people would mix that up a lot, right? That's a great exam question. See if you have your terms right. A centromere is the DNA sequence. A centrosome is the organelle, is this thing. It's composed of 2 centrioles. Take cell biology and you'll learn more about it. From this, it extends out these filaments, these fibers, that we call the mitotic spindle. Does anyone know what the mitotic spindle is? What are those filaments? What do we call those filaments? They are microtubules. Right. Take cell biology and you'll learn about microtubules in excruciating detail. Yes? Absolutely. Good stuff. Okay, so these things reach out and they make contact with the kinetochore proteins. The kinetochore proteins are connected to the centromere DNA sequence. Right? Centrosome connects to the centromere through these connections. Okay. And probably what you learned is that these guys, these filaments it's a tug of war. These guys are pulling this way, and these filaments are pulling this way, such that these guys line up at a kind of an imaginary middle of the cell called the metaphase plate. If you take my cell biology course, you'll learn it's entirely different. That's not at all what happens. And what actually happens is totally baffling, but that's the way it happens. Crazy. Take cell biology. It's my little teaser to get you into cell biology. For the purposes of this class, fine. It's a tug of war they're pulling, and they pull equally since that lines up with that. Okay. So here are the steps. I'm not going to run through them. Basically, briefly I'll run through. I'm not going to go detail. In prophase, the DNA is these long, thin fibers. Those fibers get wound up in a way that we talk about later in the course. There's a structural component that brings it together, such that they call this prometaphase. They've created a new phase since I was in college. Why? I don't know. It was so easy to learn prophase, metaphase, anaphase, telophase, PMAT. But now we have prophase, prometaphase, metaphase, anaphase, telophase. And the people who study this process just decided, well, there's some important things that happen here, so we're going to make a new phase. That's what happened. So here the chromosomes have condensed, but they're not in the middle of the cell. And so they call this prometaphase. Here the nuclear envelope's breaking apart, they're condensing. When the tug of war has gone to a point, the chromosomes line up in the middle, and the cell actually kind of like, everything in the cell just kind of halts. And actually if you look under a microscope, you can sort of see the cell actually kind of does this little like beat like that, just before the chromosomes separate. It's really interesting. But the cell actually crimps in like that for a little bit. When that happens, you know that the chromosomes are about to separate. That is, the kinetochore proteins are about to get destroyed, so the chromosomes can get pulled apart in the tug of war. When they start moving apart, that is anaphase. Anaphase is the process of them moving apart. When they reach the end, and they start getting a nuclear envelope back around them, that's telophase. And then you just need to pinch this closed, that's cytokinesis. What they've done in these images up here, these are drawings, cartoon drawings. What they've done is they've labeled the cells with different dyes. The DNA stains green, and the microtubules stain red, it looks like to me. So the DNA is diffuse, and then it breaks down into the individual chromosomes that you see because they get packed tight. Did I do that right? So blue, that is a hook stain for the DNA, and this is probably FITC for the green, and then the red would be, it looks to me like intermediate filaments, active. Anyway, so the microtubules in green, the blue is DNA. So you can actually see the chromosomes separating here in anaphase and in telophase. They're starting to form, each form a nucleus. Questions on this? Cytokinesis is the process of the cells pinching themselves closed. In plants, the plants don't actually pinch themselves closed, they just build a new cell wall between the two cells. Cytokinesis. Okay, questions on this? We've got 10 minutes to do meiosis. Okay. This is one of my favorite parts. Class. We may not get through it today. We may finish this up on Monday. I tell this story in a lot of my classes, so you may have heard it before. Before Facebook, people would create these bulletin board groups, like Yahoo bulletin board. Who's ever heard of these Yahoo forums? And then you would invite people by email to join your forum, and those people would post on there. So the way people used to keep in touch with all their high school friends is they would create the Adams High School Class of 1987 forum, and you had to be specifically invited in. And I say that one because I was in that one. I'm still in it, I think. So that's how I stayed in touch with everyone from my high school graduating class. And people would post on there, and 7 years ago, people would be posting, "Oh, just had my third kid," and everybody would. You know how you post something here, and then everybody would follow up with posts like this, and then this person would say something, and maybe piss off a whole bunch of people. You know how it would all lay out like that? You guys remember that stuff? Or is that way too old for you? Great, thank you. Anyway, people would post, "Hey, I just had my third kid, da 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 da." So finally, there's another guy I kind of hung out with. His name was Andy. And he posted on here, "Just had my first kid. I think the kid is Avery, a little girl. My wife was expecting." So, he was getting all this attention on here. I'm like, "God, I'm so jealous. I can't wait till I get to post that I had my kid." I posted on here, "Hey, Kate was born. Woo!" And people are all excited. And generally what happens is when a new one comes on, nobody's talking about Avery anymore. They're all talking about Kate. And then they go off topic and all this stuff. So this is going on for a long time. I was excited. Nobody else had a kid born for like a week or 10 days or something, so it was really a popular thread on the forum, until down here, Stacy, who I graduated with, posted that her daughter is pregnant. Kate is my first, and her daughter is pregnant. Right? So, I don't know now, I can't remember if that's a boy or a girl. But Kate is 6 months older than this child here. Stacy's, the rate of Stacy's generations is faster, right? Such that someday, that could happen, right? My daughter could marry Stacy's grandson, and they'd be about the same age, right? Stacy got started young, and I got started late. So let's think about this. If, let's say we all start with 46 chromosomes. If they contribute 46 each, now we're at 92. And they contribute 92 each. Sorry, yeah, 92. And we're at 184. But here I'm at 46 and 46 with 92. Here, we don't have an even match-up for chromosome numbers, right? So you and I are thinking, oh wow, skipped a generation here. This happens frequently in nature, where generations, there'll be a group that'll go on an island and then some of those will come back and the generations will be off like this. And the populations come back and mix and breed back together again. So in order for that type of reproduction to be successful, there has to be a way of getting back to a common number. And that's the reason, that's the value of having this meiosis, is in the process, there was 23 contributed here and 23 contributed here, so that they came back to 46. So that there were 23 contributed here and 23 here, so this stayed at 46. So that Kate has 46, to the best of my knowledge, 46 chromosomes. And that we have an equal number and this matches, and this kid will be 46, if that happens, right? So there needs to be a reduction in the number of chromosomes to a base level, and that process is meiosis. So meiosis is actually 2 cell divisions, and it starts out basically the same as a regular one. We start with a cell, we go through a G1, we go through an S phase. So we have 46 chromosomes, but there's an extra set. That is 1 chromosome, but it's 4 strands, right? So there's double the actual amount of DNA, right? So if we're at 96 and we want to get to 23, or 92, there has to be cell division back to 46 and a cell division now down to 23. So there has to be cell division number 1 and cell division number 2. So meiosis is a cell division followed by another cell division. This is meiosis 1, and this is meiosis 2. So this cell division has a prophase, a metaphase, an anaphase, a telophase, and cytokinesis. And then we have another prophase, metaphase, anaphase, telophase, cytokinesis. And one of these reduces the number of chromosomes. It's called a reduction division. I'll get to that in a second here. We may not get to it today. We may have to wait until Monday. In the first cell division is a reduction division. And what happens in that cell division is, we'll contrast this with mitosis. In mitosis, these things split apart such that this guy goes here, and this guy goes here, and this little one goes here, and this little one goes here, so that we wind up like this. The kinetochore is broken. And so we have the same, we wind up with the same number of chromosomes here. In meiosis, what happens is this guy goes over here, and this guy goes over here, and the kinetochore remains intact. We have 4 chromosomes here, 1, 2, 3, 4. And I only have 2 chromosomes here. I have the same amount of DNA, right? But our definition of chromosomes says I've reduced the number of chromosomes. Yes? The first cell division reduces the number of chromosomes in the cell. You're not interested, you guys probably think DNA content, that's not what's important. It's this artificial term chromosome that was created. Now, in the second cell division, these guys separate such that we wind up with this. This is the small chromosome, right? This guy goes here, this guy goes here, this guy goes here, and this guy goes here.
Right.
And that's basically a normal mitosis because we break the kinetic order. Okay. You've probably been through all this stuff before. I wanted to run through it again. We will talk about crossing over. Crossing over happens in prophase 1. It's a very important event. And so prophase 1 is prophase of meiosis 1. We will break prophase down into 5 more detailed steps. And we will talk about those conceptually, and in section 3 of the course, we will see physically and molecularly what happens. Okay? Thanks, have a nice weekend. Reminder, there's no class on Friday, I'm out of town. Have a great weekend.